Chapter 3, Section 4, Cellular Energy Conservation. Turns out, as I've said before in earlier sections, ATP is our drug of choice. All living cells on this planet utilize adenosine triphosphate. Okay, this is the most important energy compound, the most important energy-rich phosphate compound. There's two high-energy bonds between phosphate 1 and 2, and between 2 and 3. The phosphates 1, 2, 3 going from the adenosine out. 1, 2, 3. You'll also see it in textbooks referred to as alpha, beta, gamma. High energy there. But it is not the only high energy compound that's in the cell. Okay, there are quite a few other ones out there. Our friend phosphoenolpyruvate here, the second to the last step of glycolysis. Look at that, negative 61.9 kilojoules. ATP, our star ATP here, remember that's negative 31.8. Ones that are not going to be as quite, glucose 6-phosphate. Remember, this is the beginning of glycolysis. There's a lot of energy there. We've added phosphate there, but we haven't pushed through a lot. AMP, which is not shown here, adenosine monophosphate, means there's energy, still some energy in this carbon inorganic phosphate group bond there, but not as much as we see here in those two bonds between ATP, which would also cut there, give us a diphosphate, cut there, give us a monophosphate, AMP. We see that when it comes to high energy compounds, there's going to be a phosphate, or chances are sometimes there is a sulfur functional group someplace there that is also bringing some energy to the party. Coenzyme A, as we saw in the last slide, that one's there. It brings in some energy because it uses a thioester bond. The manufacture of ATP in many reactions is going to be based on one of three mechanisms. Substrate level. This is where an enzyme pulls a phosphate group, an inorganic phosphate, off of one thing, and then immediately attaches it to a diphosphate to form a triphosphate. You see this at the last step of glycolysis, the hydrolysis of phosphoenolpyruvate, pulling off that phosphate group to yield pyruvate. Well, that inorganic phosphate's added to ADP to make ATP. While this works good, this works great. Oxidative phosphorylation. This is what you've learned, in the mitochondria, powerhouse of the cell. This is where you use a proton motive force, the flowing of hydrogens across a membrane through an enzyme known as ATP synthase, which will take the flowing of hydrogens, causing something to move and spin. Flow of hydrogens becomes mechanical energy. The mechanical energy will then slam together inorganic phosphate and a diphosphate, and now you have chemical energy. Kind of cool. And then what is referred to as photophosphorylation, where you're going to use light to bring the energy via an enzyme to add that inorganic phosphate to a diphosphate.